Melanoma Matters is an educational resource, and the opinions we express are our own and do not reflect medical advice. If you have questions specific to your own situation, please contact your own clinician. This podcast may be available in both audio and visual, so please check your platform.
All right. Hi, everybody. Welcome to Melanoma Matters. I'm Sapna Patel.
I'm James Larkin. Hi.
Today we have a very special guest joining us from Denmark, Inge Marie Svane. Inge Marie, thank you so much for taking time out of your schedule for joining us.
Welcome.
So perhaps we could get to know you a little bit. Could you tell us about your maybe background, where you grew up, where you did your studies, and how you came to the position that you're in now?
My interest in cancer immunotherapy started out already when I was a medical student. So I've been working with this area within cancer therapy for more than 25 years. I made a basic PhD at the university in immune surveillance in immune model, nude mice and so on, where I kind of documented that it matters whether you have a T-cell system or not. So it matters how the tumor grows up and how the antigenicity of the tumor are. But then I moved out to the clinical world, and I started treating patients, and then my dilemma started out because I actually liked the interaction with the patients very much. So I decided not to go back to the university, and instead I tried to establish my own research group. At the same time, I was trained as a specialist in oncology. So it took quite some years to do my training because I worked all the time also in the lab myself, and then I started a group, like translational, you could say. But some years ago, in 2006, I think, we started out a group where we joined. I was joined by a more basic research group, and we established this Center for Cancer Immunotherapy in Denmark. So it's a long time ago now, but we have been growing since then. We were like 15 people when we started out, and now we are around 90 people. So I'm also a director of that center, but I have continued to work in the clinic all the time. And in the clinic, I have responsibility for melanoma care. So when I took over the area of melanoma, it was like we didn't have really anything, but I established the high-dose interleukin 2 at that time. And then we had like the temozolomide. That was the only thing. And I think it was only a couple of years after it all took off, and we had the checkpoint inhibitors and so on. So it has been a great thing to actually work with melanoma patients, and at the same time developing clinical trials, testing our own immune therapeutic products from our center side. And that has been focused on cancer vaccines, and then lately very much on the T-cell therapy.
Yeah, that's great. I mean, I still unfortunately remember the days of temozolomide. I know Sapna does as well, so it's been amazing. I was at a patient advocacy meeting at the weekend, and what I was asked to talk about was the other 50%, meaning the people who don't get durable benefit from checkpoint inhibitors. And obviously one of the things I talked about was cellular therapy. But before we get into that in detail, I'd love to hear your historical perspective on cellular therapy for melanoma, because it's not new, right? But it's just, I guess, in the last year or two, it's something we're talking about a lot more. But how do you see that? Because I guess it's been around for decades, really, hasn't it?
Yeah, for several decades. I mean, the work from Steven Rosenberg's group has been amazing, and we have known that for actually many years. But I think it has been a struggle to move it forward because it's not an easy therapy. You need a lot of technical experience in the lab to be able to produce and manufacture the T cells, combined with the clinical setup with a very complex therapy and very intensive one also, with the high-dose chemotherapy and the high-dose interleukin 2. So I guess that's some of the reasons why it didn't spread out more fast than it actually did. So the interest from the commercial side was extremely low until like Iovance grabbed it and ran. And it is amazing that so few centers around the world actually picked it up. We actually visited NCI at the time when we decided to put it up. And we started out in head and neck cancer because we thought we should do something else, and it wasn't there. But then it was very difficult to work with the head and neck cancer patients because they are not in very good performance status when they are disseminated. And we then decided to try to kind of establish it in melanoma. And since then, we have been working quite a lot with that in melanoma. And it is, I think, as you say, it is a very important point that you can actually benefit from T-cell therapy independently of whether you had a checkpoint inhibitor refractory disease. And that's so important because these are the patients that we have now that we do not really know what to do about and how to treat them.
Yeah, so that would be almost my biggest question. I mean, we've got data from Lifelusa and the trial that you did with John Hannon, randomized trial, very important trial versus IPI. So we can see those efficacy signals. But where do you see the place of cellular therapy in melanoma, say, in 5 years' time in terms of lines of therapy? Have we got a way we could select people out or enrich the people, say, in first line who we think might benefit?
Yeah, and I think it's an important point that these are really highly selected patients, and we need to remember that it's not for all comers. First of all, you need to, I mean, the age does actually matter. So we're not treating patients above 75 because it's to tolerate this very intensive chemotherapy and IL-2. It's really tough, and you can, I mean, you can be life-threatened by the side effects. So I think that's important, but also actually that you need to be able to resect the tumor lesion without really hurting the patient. And that's not for all melanoma patients. Even though the melanoma frequently start in the skin, when it's in oncology setting, you normally have the most disseminated disease inside, and it can be a rather tough thing to go for melanoma metastasis in liver or lung. So there are a lot of limitations on that in regard to which patients to treat. My opinion at the time being, I think that still, even though we do not have the same kind of long-term side effects, chronic side effects from T-cell therapy as we can experience from checkpoint inhibitors, I still think that the total level of side effects are lower with checkpoint inhibitors, so I really would go for checkpoint inhibitors as first line. But there are many of our patients where we do not treat with this combination. Maybe, I mean, because it was a low disease burden, or in Denmark at least we also use the PD-L1 marker. And for those patients, it would be more obvious, I believe, to actually move forward with T-cell therapy if they progress on anti-PD-1 than to try to go for another checkpoint inhibitor. So I think it could be in that setting, and I think from the Iovance study as well, you have seen that for all late lines, it's around, I mean, 36% response rate. But if you go for the patients who have progressed on the ipi-nivo combination, it's a bit lower. Yeah.
I wonder if I can ask you a question, Inge Marie, about as we're now using cellular therapy as a potential standard, second line or beyond, what do you feel is the next obvious step for us? Should we be genetically modifying our TIL and having them express shutdown signals, suicide switches? Should we be giving them knockout receptors for TGF-beta? Is it the interleukin-2 portion? Should we do something to adjust that interleukin-2 so that, you know, some groups like IL-15, some groups like IL-21 because they promote more of a memory cell phenotype? Or do you think it's from that very first step, the tissue procurement, getting tissue from biopsy specimens as opposed to a surgical specimen, reducing anesthesia time, you know, quantity needed of tissue, that kind of thing? Where do you see us? Potentially we can make progress in all of these, but where should we be focusing?
I was about to say the same thing, and I think we need to move forward in all directions, right? There's a lot of things that we can optimize. I mean, both in the way that we prepare and manufacture the T cells, we know that. And we know that we lose a lot of activities when we move from the early cells that we produce, like the young cells, and then moving through the very large expansion in the last couple of weeks, we lose a lot of specificities. We can see that the clonality, it goes down. And for sure, we are losing some of the important specificities, I'm sure. So we could probably do a lot about that, how we kind of start the initial manufacturing of the T cells. And then in regard to how we could kind of manipulate them, I also think we have a lot of possibilities. So we are working on a couple of the directions here. So we're just starting up with a clinical trial now, which has been approved, where we are kind of CRISPR knock out the PD-1 expression on the T cells. You could say you could achieve the same thing maybe by combining it with an anti-PD-1 antibody. But I think what is also important here is that we are only kind of manipulating the T cells, which are concentrated around the tumor activity. So that should hopefully mean that we see less side effects than if we combine it with a checkpoint inhibitor. Because if you look at one of the things that we actually do not that frequently see for T cell therapy, that's the autoimmune side effects. It's very rare that we see that. So in that way, there's not a lot to boost by knocking out the PD-1, right? So we really truly believe that we can maybe have a more focused, continuous activation of T cells reacting against the tumor cells by knocking out the PD-1 on the cells. But other options are, as you said, also to introduce something, and we have also another trial working on introducing IL-7, but in also, again, a very focused way, where it's only when the T cell receptor interact and get activated by the antigen intratumorally, then it starts producing the IL-7. And that is was meant to be like, to protect the patient against the side effects. Because I think in the setting of T cell therapy, we can't really tolerate more side effects, right? It's so intensive for these couple of weeks when they're hospitalized that this is enough. Yeah. So whatever we do, it also has to be taken into considerations that we need to hopefully lower the side effects, but at least not increasing them. So we work together with in a European setting. We have a EU grant. It's from also from Barcelona, where they have established a trial where we are trying to use another kind of IL-2. A kind of IL-2 which is again not activated in the same way systemically, in the hope that we can reduce the IL-2 side effects, because I would say they are actually the most severe ones. So I hope that we also in the future will be able to work on the dosing and how maybe new kind of IL-2 and maybe new kind of cytokines, which can reduce the side effects, but at least also continue the same efficacy.
Yeah, I mean, that all sounds great. I mean, one of the things I've been wondering a little bit, and I agree with what you were saying, that probably for a lot of patients, you're still going to want to go with a checkpoint inhibitor first of all. And if it's single agent, it's probably going to be well tolerated. And if they're sensitive, it's great. They might not need much more treatment. But I wonder, and neoadjuvant might be a bit of an example of this, but also metastatic disease, whether we should be thinking to ourselves quite early on, is this a patient who's really going to get durable benefit from a checkpoint inhibitor or not? Yeah. Rather than waiting for multiple lines of therapy, and then you've got someone who's been on steroids and they're immunosuppressed and they've got brain mets. But actually, in our heads, if you like, treating metastatic disease, let's say in the first 6-12 months, and you know those data from various sources that where quality of response to checkpoint inhibitors will correlate quite tightly with long-term outcome, but whether we should say to ourselves, well, actually, this isn't a good enough response to a checkpoint inhibitor quite early on, maybe even a neoadjuvant. That's where we should be trying to understand more about the place of cellular therapy, because I think for that kind of patient, they're probably going to be easier to treat in lots of different ways, and I think you can justify then the potentially greater toxicity as well from cellular therapy because they failed or they're going to fail checkpoint inhibitors. Do you think that's plausible, or it's just me sort of, being optimistic about which way we're heading?
No, but I agree with you, and I think that is a whole issue about biomarkers as well. because they are going to hopefully help us out in the future. And I mean, there's a lot of assessments of biomarkers, but all of it is kind of explorative. And I think we are completely lacking the really solid and validated markers, and probably we will be working with profiles in not one marker, but of course profiles in the future, and that will hopefully help us out and help the patient out, so that we're not offering them combination checkpoint inhibitors if we can see that they will have a very, very small chance of efficacy, and instead move them forward with the T-cell therapy. And I think like for other kind of treatments and later line, the lower efficacy rate, and it's the same with T-cell therapy. We know it can be extremely difficult to kind of have the patient ready and then not progressing too aggressively after BRAF/MEK inhibitor. Waiting for the T-cells can be extremely difficult. So in Denmark here at the moment we have this approval of what I call an early access program, but it is per patient based. So I have to apply the authorities for allowance to give the T-cell therapy. And we have actually argued that this would be also feasible for patients who have not been treated with BRAF/MEK inhibitors, because afterwards it can be like impossible. And as you said, they can probably, they turn out to have brain metastasis, and you have a lot of issues there.
Yeah, well, it's good. I mean, I think we have the same experience as well, certainly.
Yeah.
Working with lifileucel and Iovance. So that sort of brings me slightly to another question, and from what I can gather, you and John Hannon as well, I think, have access to cellular therapy and reimbursement, correct? And that's a struggle. So I mean, Sapna in the US, lifileucel's got FDA approval. Do you think that kind of the regulatory and reimbursement world, you know, more widely in Europe, it just feels a little bit like it's not kind of set up for this kind of thing very well, and it's a real struggle? But, you know, and you're really close to this. I'd love to hear your thoughts.
Yeah, we can cross fingers, and we've been struggling with this for like, I think it's almost 2 years now. Yeah, no laughing. No, the system is completely untrained for academia, I would say. Everything is set up for the industry, and it's endless, the challenges that you're meeting, because you do not have the competences yourself, and you do not have really the right people to help you out, because nobody has tried to move forward as an academia towards EMA with this a submission of a market approval application. So even though John Hannon and Kai has actually been able to find a consultancy, they haven't got the experience, right? So of course they're helping us out in the level that they have, but I mean, nobody has tried it before. And in EMA, they are also challenged by the lack of experience. And there's a lot of discussions actually going on in Europe at this time point. I've been approached by, they have made a committee around this ATMP, the advanced cellular therapies as well, as a gene therapies, and they try to kind of dig into also the hospital exemption, because it's the way that it's being used in the different countries in Europe is completely different. So it's interpreted very strictly, for example, in Denmark and many of the countries in Europe, but in Spain, they are allowed to do a lot of things like the CAR T cells. They do their own CD19 CAR T based on this hospital exemption, and they have now the second approval, but only regionally, like in Spain. And at the same time, it was declined in Denmark. I was declined applying for hospital exemption, while John Hannon in the Netherlands was approved based on the same data, right? So in that way, it's very difficult. And the whole discussion also about competition. Is this a fair competition to the companies, or is this really unfair? And how should we put the price on it? Yeah, there's a lot of things here. So I think within the next, it will be very interesting to see within the next 5 years where we will end up. Where the politically and regulatorily, and also, I mean, like the collaboration with the industry, I think there's room for all of us. That's really how I see it, because we have so many patients around Europe and around the world who need this therapy, and it's not near to covered by the industry at the moment. So I think there's a lot of space that we can just try to help each other. And I think also it's a healthy thing to have competition also when you're a company. So it's like the discussion of this point of care production as compared to a centralized production also. And I think we will have room for both ways to do it, I believe. And also to secure actually the continued development and research in T-cell therapy. You need to have it right next to the clinical researchers and the academia.
Yeah, it just feels to me, I mean, I know how frustrating it's been, at least I think I've got some idea of how frustrating it's been, that you have a therapy that can really help patients durably. And there's all these other considerations that are coming into it, but at the end of the day, this is something that really can make a difference to patients. So, congratulations to you and John and everybody else on really, pursuing this. But it is a point, I suppose, isn't it, that there's all this beautiful clinical and scientific work, but actually then to deliver it to patients afterwards, there's a whole load of other considerations. So listen, this is going to be my final question, Inge Marie, and Sapna may have to ask other things, but based on what we've discussed, where do you see the prospects for this type of strategy in other solid tumour types? I mean, there's data certainly from Iovance and others in various other settings. Do you think it's going to be a sort of a mainstream treatment in other solid tumour types, in appropriately selected patients, obviously, not for everybody? Do you see the promise there?
Yeah, I do when you say in selected patients. I think it's very encouraging data on head and neck cancer and also lung cancer. But again, in lung cancer, you need to be able to go through a resection of the lung metastasis typically. And many of the head and neck cancer patients, they are really not in a performance status where you can drag them through the high-dose chemotherapy and IL-2. So we need to figure out how to, increase the efficacy in those patients and decrease the side effects for sure. And I think for that, it was important initial study that they tested it in combination with checkpoint inhibitors as well. And you can also imagine like genetic modification can also improve it. And maybe what we're also working, this is like really preclinically, but we're also working on a CAR TIL. So you actually kind of introduce a CAR in the T cells during manufacturing. So you have a very potent target, and then you combine it with the broadness of the repertoire of the cells. And the combination of that could maybe lead to a higher efficacy clinical also. I think so. We have so many different very exciting tools for manipulating those cells. So I think there will be a lot of new strategies out there in the next years.
Okay, Sapna, anything else from you?
No, I think that was incredible. I mean, we even only just talked about a portion of your research, cell therapy. We didn't even get the vaccine part, and there's so much, I think, here coming. So exciting to see.
Welcome.
And of course, congratulations to you and John and everybody there that's working on this cellular therapy in Northern Europe. I only know, just because it's a pretty monumental date, it was 2 years ago yesterday that your New England Journal of Medicine paper came out. As you're saying, here we are 2 years later. Where are the approvals and how do we get this to patients? But thank you so much for coming on our podcast.
Welcome. It was really great talking to you.
Wonderful.
Take care. Bye.
Bye.